Hi everyone, here comes the next topic of my lecture, which is cell cycle and regulation. First, let's go through the specific learning objectives. So after this lecture, you should be able to understand and describe the eukaryotic cell cycle process, discuss the regulatory mechanism of the cell cycle, and then discuss briefly about apoptosis and cancer. So far you have learned about the cell, particularly cell membrane and its organelles, also about DNA synthesis, which is the replication. So this topic on cell cycle will be the meeting point of those two previous topics, whereby DNA replication occurs during a phase in the cell cycle before the cell divides in another phase of the cell cycle. But before that, let's recap on mitosis and meiosis first. As you have learned before, mitosis is to preserve the diploid number of chromosomes over repeated generations of cells, while meiosis is to maintain the diploid number of chromosomes over repeated generations of organisms due to the haploidy of the gametes. Thus, cell division in the cell cycle refers to mitosis. If you can remember a session of your biology practical class years back, whereby you observe the stages of mitosis in onion peels, so this here is an upgraded version of what you saw under the microscope before, which is an immunofluorescence micrograph. Here, fluorescence were used to give distinctive color to the different structure of the mitotic spindle. So we can clearly see the arrangement of the spindle from prophase until telophase. Alright, enough recap. Let us continue with the cell cycle, which refers to the orderly sequence of events involving duplication of the cell contents and then division into two, essentially to produce two genetically identical daughter cells. It begins with the faithful replication of DNA in each chromosome of daughter cell to produce two complete copies. Then, the replicated chromosomes is accurately distributed or segregated to the two daughter cells, each receiving copy of the entire genome, ultimately producing two genetically identical daughter cells. In addition to duplicating their genome, most cells also duplicate their other organelles and macromolecules, which is important because otherwise daughter cells would get smaller with each division. Therefore, the basic function of the cell cycle is to duplicate accurately the DNA in the chromosomes and then segregate the copies precisely into two genetically identical daughter cells. However, it is worth to know that not all cells undergo cell division. Essentially, these three types of cells. The highly specialized cells, for example, the nerve cells, muscle cells or red blood cells which have lost their ability to divide, cells that normally do not divide but can be induced to do so upon stimulation, for example, the liver cells and the lymphocytes, and cells with a relatively high level of mitotic activity. The cell cycle has four phases, which are the S phase. S refers to synthesis of DNA or replication, the M phase, which is mitosis, and two gap phases known as G1 and G2. So most cells require much more time to grow and double their mass of proteins and organelles than they require to duplicate their chromosomes and divide. Thus, partly to allow time for growth, most cell cycles have gap phases. We have the G1 phase between M phase and S phase, and G2 phase between S phase and mitosis, which might occupy 23 hours of the 24-hour cycle, leaving the remaining 1 hour for the M phase. Thus, cell growth occurs throughout the cell cycle except during mitosis. However, essentially the cell cycle consists of two major phases. Chromosome duplication that occurs during S phase, S for DNA synthesis, which requires 10 to 12 hours, and the M phase for nuclear division or mitosis, that is, the copied chromosomes are distributed into a pair of daughter nuclei cells, followed by cytoplasmic division or cytokinesis when the cell itself divides into two. So the S phase occupies about half of the cell cycle time in a typical mammalian cell, while the remaining hour or so is for the M phase. As I have mentioned earlier, the gap phases provide time for the cell to monitor the internal and external environment to ensure that conditions are suitable, and also to ensure that all preparations are complete before cells commits itself to the S phase and mitosis. In fact, the G1 phase length can vary greatly depending on external conditions and extracellular signals from other cells. For instance, if extracellular conditions are unfavorable, cells delay progress through G1 and may even enter a specialized resting state known as G0 or G0 that can remain for days, weeks, or even years before resuming proliferation. If extracellular conditions are favorable and signals to grow and divide are present, cells in early G1 or G0 progress through a commitment point near the end of G1, known as the restriction point or start. But after passing this point, cells are committed to DNA replication, even if the extracellular signals that stimulate cell growth and divisions are removed. Next, we shall look into the cell cycle regulation, which actually refers to a system of switches with many important features, essentially to increase the accuracy and reliability of the cell cycle progression. The switches are generally binary, either on or off, to launch events in a complete irreversible fashion. They are remarkably robust and reliable with backup mechanisms and other features, allowing the system to operate effectively under a variety of conditions, and even if some of the components fail. And finally, the system is highly adaptable, thus can be modified to suit specific cell types or to respond to specific intracellular or extracellular signals. The cell cycle is regulated at three key transition points based on the chemical signals reflecting the cell's intracellular and extracellular state. The first key transition point is the restriction point or the start point that detects presence of extracellular growth factor. If passed, cell commits to cell cycle entry and thus replication. The second one is the G2M transition point. This is a control system that triggers the early mitotic events towards chromosome alignment on the mitotic spindle in metaphase. This transition point can be indefinitely arrested at the end of G2 if cell division is not necessary. But at late G1 and G2 phase, the cell can exit from the cell cycle and enter a non-dividing phase known as the G0. The third key transition point is the metaphase-anaphase transition, which is a control system that stimulates sister chromatic separation towards the completion of mitosis and cytokinesis. This is when all the chromosomes are properly attached to the spindle. Next is the progression through the cell cycle, which is controlled by cyclin-dependent kinases, CDKs. CDKs are the key regulators of the cell cycle. It exists as a complex of two polypeptides. One binds to ATP, another one having a kinase activity. The activities of these kinases rise and fall as the cell progresses through the cycle. That refers to cyclical changes in the phosphorylation of intracellular proteins that initiate or regulate the major events of the cell cycle. However, CDKs are only active when complex with cyclin proteins, whereby one CDK can bind with different cyclin. So we have G1 cyclins, G1 CDKs that is active at the G1 restriction point. This S cyclins CDKs that is active at the S phase point, and the mitotic cyclins mitotic CDKs active at the G2M transition point. There are actually four classes of cyclin. The first one is G1S cyclins, which activate CDKs in late G1 that help trigger progression through start, resulting in a commitment to cell cycle entry. Their levels falls in S phase, whereby S cyclins bind CDKs soon after progression through start and help stimulate chromosome duplication. The S cyclin levels remain elevated until mitosis. Meanwhile, the M cyclins activate CDKs that stimulate entry into mitosis at the G2M transition, and M cyclin levels falls in mid-mitosis. Finally, is the G1 cyclins that helps govern the activities of the G1S cyclins, which control progression through start in late G1. But essentially, there are three main cyclin CDK complexes, which are G1 cyclins, G1 CDKs at G1 restriction point, the S cyclin CDKs at S phase, and mitotic cyclins mitotic CDKs at G2M transition point. The next two slides will be on the mechanisms of CDK actions, and I will discuss only two main pathways here, but you can read the rest in the textbook. So firstly, when mitotic CDK and mitotic cyclin bind together, they form an inactive complex whereby two inhibitory phosphate groups are attached to the CDK molecule by enzymes called inhibiting kinases. An activating phosphate group, labeled in yellow, is added by an activating kinase, but the CDK remains inactive as long as the inhibitory phosphate groups labeled in white are present. So, a phosphatase removes the inhibiting phosphates, thereby activating the mitotic CDK cyclin complex. Therefore, activation involves addition of inhibiting and activating phosphate groups with the removal of inhibiting phosphate by phosphatase, giving a positive feedback. After activation of mitotic CDK cyclin, its protein kinase activity triggers the onset of mitosis. Secondly is how the G1 CDK cyclin regulates progression through the restriction point by phosphorylation of the RB protein. The RB protein controls the expression of genes that code for protein needed to pass through the restriction point or the start point. In dephosphorylated state, the RB binds to the E2F transcription factor, thus prevents the E2F from transcription of genes coding for protein needed for replication. When cells are stimulated to divide by the growth factors, RAS pathway is activated, thus G1 CDK cyclin is activated. RB protein is then phosphorylated thus can no longer bind to the E to F, allowing E to F to activate gene transcription, then synthesis of proteins needed to trigger the onset of S phase. Another regulation for cell cycle is replication licensing, which is to ensure that nuclear DNA molecules undergo replication once and only once prior to cell division. It means that after the DNA is replicated at any given replication origin in S phase, the DNA at that site does not become competent for a further round of DNA replication until the cell has first passed through mitosis. The license is provided by origin replication complex, the helicase loaders, and mini chromosome maintenance MCM proteins. The licensing is turned off at the end of G1 by the production of CDKs and or germinin. In summary, passage through the three main transition points in the cell cycle is triggered by protein complexes made of cyclin and CDK, whose phosphorylation of other proteins induces progression through the cell cycle through these three checkpoint pathways. First is the mitotic spindle checkpoint, to prevent anaphase chromosome movements from beginning before the chromosomes are all attached to the spindle. A wait signal inhibits the anaphase promoting complex so that no separation of sister chromatids happens until all chromosomes are properly attached to the spindle. The DNA replication checkpoint is to monitor the state of DNA replication, that is to ensure DNA synthesis is completed before the cell exits G2 and begin mitosis, which is the action of mitotic CDK cycling. The DNA damage checkpoint is to prevent cells with DNA damage from proceeding through the cell cycle until the damaged DNA is repaired. Halting of the cell cycle at different points that are late G1, S, and late G2 by inhibiting different CDK cycling complexes involving the role of p53 protein, which is known as the guardian of the genome, that we will look into in the next slides. Therefore, the checkpoints are to ensure error-free completion of DNA replication before entry into mitosis and to maintain temporal coordination of S and M phases, whereby the cell cycle will be arrested at specific points in response to different conditions. The cell cycle will be arrested at G1 in response to DNA damage, arrested at S phase in response to DNA damage as well as incomplete replication. It will be arrested at G2 point in response again to DNA damage, and finally it will be arrested at mitosis in response to unattached kinetochore. In summary, herewith is the general model for the cell cycle regulation. The G1 CDK cycline acts at the restriction point or start by phosphorylation of the RB protein. Mitotic CDK cycline acts at the G2M boundary through phosphorylation of proteins for chromosome condensation, nuclear envelope breakdown, and spindle assembly. And the same mitotic CDK cycline also influences metaphase-anaphase transition via phosphorylation of anaphase promoting complex, which then triggers chromosome separation. And finally, we have the checkpoint pathways that monitor the cell for DNA damage, DNA replication, and chromosome attachment to the spindle. All of these mechanisms working in sync to regulate the cell cycle. Thank you, but please stay tuned for part 2 of the cell cycle and regulation. We shall now continue with part 2 of the cell cycle and regulation topic. But first, we need to know about DNA damage. DNA damage are alterations in the DNA that arise either spontaneously or from exposure to DNA damaging environmental agents. Most DNA damage is repaired shortly after it occurs, hence does not affect future generations. Spontaneous DNA damage include depurination and deamination. Depurination refers to loss of either adenine or guanine via hydrolysis, while deamination refers to loss of amino group from either cytosine, adenine, or guanine. Cytosine is most susceptible to deamination, which is converted to uracil. DNA damage by environmental mutation includes formation of DNA or bulky adducts due to exposure to chemicals mutagen, such as the base analogues and base modifying intercalating agents. Formation of pyrimidine dimers also occurs due to exposure to radiation of UV rays, which block the replication and transcription. The X-rays also cause ionization that leads to formation of highly reactive intermediates. I will talk more details on DNA damage and the mechanism of repair in the next lecture entitled DNA damage and repair. I have mentioned in part 1 of this lecture about P53, known as the guardian of the genome, most likely due to its role in response to DNA damage. DNA damage activates the ATM or ATR, which then in turn activates the checkpoint kinases that leads to phosphorylation of P53 proteins. Phosphorylated P53 will then bind to DNA and activates transcription of gene coding for P21 protein that will inhibit the CDK cyclin, thus prevents phosphorylation of RB protein. Hence, cell cycle is arrested at the restriction point or the start to allow for repair of the damaged DNA. If DNA damage cannot be repaired, the genes coding for proteins that trigger cell death or apoptosis will be activated, and in turn it will activate PUMA that will promote apoptosis by binding to BCL2, thus blocking its action as apoptosis inhibitor. So far, you have learned about the eukaryotic cell cycle process and the regulatory mechanism of the cell cycle. So in this part 2, we will discuss briefly about apoptosis and cancer. Cancers develop by stepwise accumulation of mutations involving oncogenes and tumor suppressor genes. Genome sequencing revealed mutations in approximately 50 to 75 protein coding genes found in breast, lung, or colon cancers. Common mutations involve conversion of proto-oncogenes into oncogenes and inactivation of tumor suppressor genes. Proto-oncogenes encode proteins that are growth factors, growth factor receptors, signal transduction proteins, cell cycle regulators, and regulators for apoptosis. For instance, the protein that control progression in the cell cycle through the restriction point or the start point, as we have learned before, known as RB protein. However, mutations in proto-oncogenes converts them into oncogenes, which code for proteins that stimulate excessive cell proliferation and or promote cell survival by inhibiting apoptosis. For example, a small change in DNA sequence, such as point mutation or deletion, may produce a hyperactive protein when it occurs within a protein coding sequence, or lead to protein overproduction when it occurs within a regulatory region for that gene. Gene amplification events, such as those that can be caused by errors in DNA replication, may produce extra gene copies that can lead to overproduction of the protein. A chromosomal rearrangement events involving the breakage and rejoining of the DNA helix may either change the protein coding region, resulting in a hyperactive fusion protein. Next is the tumor suppressor genes, which are genes that encode molecules or proteins involved in the regulation of cell proliferation. It has a normal function to inhibit cell proliferation in response to certain signals such as DNA damage. Thereby, they affect cell cycle regulation, signal transduction, transcription, and cell adhesion, whereby products of tumor suppressor genes frequently modulate pathways activated by products of proto-oncogenes. An example of an important tumor suppressor gene is the p53 gene that codes for p53 protein, which we have seen earlier on its role to protect the cell cycle against DNA damage. Hence the nickname guardian of the genome, whereby p53 stops replication in cells suffered from DNA damage and targets any unrepaired cells to apoptosis. However, tumor suppressor genes contribute to the development of cancer when both copies of the gene are inactivated, whereby almost half of the 10 million people diagnosed with cancer has tumors with p53 mutations. An example is when cells exposed to UV rays or chemical mutagen, it induces DNA damage, which will then stimulate the p53 pathway that will trigger cell cycle arrest and apoptosis, hence prevent proliferation of cells with DNA damage. However, this protective mechanism is missing in cancer cells because of the mutations disrupting the p53 gene that will lead to inactivation of the p53 gene, thus failure of apoptosis, hence reproduction of cells with DNA damage. Other examples include mutation of p53 gene found in lung cancer caused by carcinogens in tobacco smoke, and the UV signature thymine dimer mutation in skin cancer due to exposure to UV sun rays. p53 protein can also be altered by cancer-causing virus. For example, the human papilloma virus HPV has the oncogene that codes for E6 protein that will direct the attachment of ubiquitin to p53 protein, hence disrupting the p53 protein. Another important tumor suppressor gene is the RB gene, which was discovered in young children with hereditary retinoblastoma due to deletion in the specific region of chromosome 13. The RB gene codes for the RB protein that has a role in controlling progression from G1 to S phase in the cell cycle, as how we have learned in the first part of this lecture. Mutation in the RB gene removes the restraining mechanism that can lead to uncontrolled proliferation. Like the p53 gene, the RB protein can also be altered by cancer-causing virus. The human papilloma virus, again, has an oncogene that codes for E7 protein, which can bind to RB protein of infected cells, thus prevents its role in restraining cell proliferation. Finally is the apoptosis or programmed cell death, which is a regulated energy-dependent sequence of events by which a cell self-destructs to remove superfluous or unwanted cells. Apoptosis is actually a normal process in a complex organism during embryogenesis, maintenance of proper cell number in tissues, removal of infected or injured cells, maintenance of immune system, as well as aging process. It can be initiated by external signals recognized by the death receptor tumor necrosis factor TNF, deprivation of growth hormone, as well as irreparably damaged DNA. However, failure of apoptosis to remove excess or damaged cells can contribute to the development of cancer. So that was the end for the topic cell cycle and regulation. These are the list of references, and you can email your question to the email stated here. Thank you.